Okay, class, today we are picking back up with chapter 4. We will finish chapter 4 today and start chapter 5. This lecture serves as the lecture for January the 27th, and we are going to be starting out with lysosomes. So, lysosomes are specialized vesicles within animal cells. So, lysosomes are going to contain these digestive enzymes, and they use these enzymes to break down large biomolecules and even sometimes worn out organelles within the cells, such that you can break those down and reuse the pieces, or actually exocytose the material from the cell if needed. So these are kind of like the garbage men of the cell. So they go in, they break things down using these digestive enzymes, and they do this through a process of phagocytosis. So if we look at this image here, you can see this, let's just call it a food particle. That's what they're calling it here. It could be a food particle of some kind that needs to be broken down. It could be actually another organelle within the cell that is breaking down, and it needs to be digested. So what it does is this invaginates out on the outer membrane of the lysosome and completely engulfs and surrounds that particle, and then buds inward, forming this food vacuole. Okay, that's a vacuole again is just an encapsulation of something within the cell. So the lysosome digestive enzymes, as you can see here, come and actually form this um, phagocytic lysosome pocket where it comes in and digests the food particle, and then as it breaks up, it can exocytose that from the lysosome vesicle, and then it's going to contain all of this material that just gets spit back out into the cell. So there are pseudopods usually located on lysosomes, and these help entrap things that it needs to digest. So they're kind of like little tentacles, but we call them pseudopods. It also helps with its motility and moving around. All right. So, what is the cytoskeleton of the cell? Cytoskeleton is actually a network of protein fibers within that have several functions. So, they're going to help maintain the shape of the cell. They're going to hold some organelles in specific positions. And then they allow the movement of the cytoplasm and vesicles within the cell. So, we actually use the cytoskeleton to not only help maintain the shape, but allow for the movement of the cell itself and the cytoplasm within it. So it also is just going to enable that cell to within multicellular organisms to move around. So here are the components of the cytoskeleton. So they are the microfilaments, the intermediate filaments, the microtubules, and then each is going to have different size and different functions. So as you can see in this picture, if we were to blow it up right here, located on the outside of the cell is the plasma membrane, and then located usually most closely to the outside of the cell are these microfilaments, and then the intermediate filaments are these a little bit larger. That's why they're called intermediate. They're the middle size of the tubules of the cytoskeleton, and they're going to be more towards the middle, and they help form some structural components. And then you got these microtubules that are much larger, and they're going to help with some transport of different things. So microfilaments, the very smallest one, they're involved in movement, so they actually allow for the movement of the cell. So that's why they're located closer towards and throughout like the structure towards the outside, and then form these bridges that actually help support and then allow for it to contract and expand and allow for movement. But they determine the size and stability and the shape of the cell. And they're going to be made from these actin monomers. So each one of these dots is an actin subunit or a monomer strung together to form actin filaments. Okay, so those are what comprise microfilaments. Are the actin monomers strung together to form actin filaments. So this is exactly we see these a lot in muscle cells, especially. But this is exactly kind of what it looks like. So you have this actin filament, and then you've got these myosin head groups, and the myosin head groups actually latch on and cause this contraction so that it actually brings the fibers together and forms this contraction. So that's how the cell is able to contract, and then when it releases, it expands back out. So those microfilaments are useful in movement and the shape of the cell like that. So they're going to really stabilize that shape and allow for the movement. Now, microtubules are going to provide a framework for motor proteins to move structures within the cell. So they're like the highways. And you can see here that there are these subunits that coil around to form a tube. But they're made of these tubulin dimers. It's 13 chains of dimers actually surrounding a central cavity, and that is the microtubule. Okay, so what are cilia and flagella then? So we've talked a little bit about the motility of cells, and cilia and flagella are highly used in that processes. So cilia and flagella are an ultrastructure. They're going to have the same 9+2 array of microtubules. So they are kind of comprised of the same structure, but the biggest thing that keeps them separated is that cilia are shorter and more numerous. So you can see, give me 1 second. So you can see here that the cilia on this cell right here it's kind of like, looks like little hairs. So anytime you see a cell that is just covered in a hair-like material, that's most often going to be a cilia that is allowing for motility of that cell. And then flagellum or flagella are these long tentacle-like structures found on a cell and are much fewer than you will see with cilia. When you see cilia, there's hundreds, if not thousands of them, where flagellum are going to be more singularly situated, like in this cell here. So there's going to be a difference in beating pattern. The flagellum is going to use this propeller-like motion that allows it to propel itself. And then the cilia uses a whipping back and forth motion, like a beating pattern, to actually move it, allow for movement of the cell. So, but they're both made similarly. They have these same array of microtubules. So you also get these dynein proteins that are going to be stringing them together and holding everything together. And then, these radial spokes here are same. So the whole construction of the cilia and flagella are similar in that they're made the same way. Okay, so then there are those intermediate filaments, the middle size that we talked about now. So they're going to range in diameter from 8 to 12 nanometers. They're larger than the microfilaments, but smaller than the microtubules. And they're going to be important for cytoskeleton fixtures they're a more permanent fixture, in other words. So they support the shell's shape and they fix organelles in place. Okay, so where we saw the microfilaments up here are really important for shape, we saw determine and stabilize shape. They are much more fluid in movement. They can move. And this is seen in muscle cells with these microfilaments. They're going to be used in moving and contracting the cell and that kind of thing. Whereas these intermediate filaments are much more permanent in their cytoskeleton fixture. And so they are going to give the cell its shape, but also they fix organelles in place. So they kind of hold the organelles where they're supposed to be. All right, so extracellular structures are also important within the extracellular matrix. And these are going to help organize and connect cells together. And so there are three main components of the extracellular matrix. They're going to be collagens and other fibrous proteins. You're going to have glycoproteins called proteoglycans. And then you're going to have these linking proteins called integrins. So you can see here in this picture that this area on the outside here is the extracellular fluid, and that's where you can find the extracellular matrix made up of these three main components. So you can see the collagen, you can see the fibronectin, um, uh, and then which are like lectin proteins and that kind of thing. And then you're going to have these integrin proteins, um, which are comprised of these things as well. So that is the extracellular matrix. All right. So what about extracellular structure in plants? So the plant cell wall is what gives it support. It's going to give a barrier to infection, and we're going to use plasmodesmata to connect the cells. So you can see the plasmodesmata here linking these cells like little bridges between the cells. So those are all plasmodesmata. Okay. They link plant cells together. That's important to remember. They're going to kind of connect the plasma membranes of the two separate cell walls. So this right here is one cell, and then this right here is another cell that is being linked together. All right, so intracellular junctions, they're going to provide direct channels of communication between the cells. And then plants and animals do this differently. So the plasmodesmata actually are what link the cells together in plants. And they also are going to allow for these channels to allow information and ions and different things to pass between them. They connect the cytoplasm and allow for that material to move from cell to cell. Now we have tight junctions. Now tight junctions are watertight seals between animal cells that prevent material from leaking between each other. So they're going to be basically just sealing the cells together in their tight junctions, meaning nothing can get through it. They're found in epithelial cells that line internal organs and cavities. And they are very, very important. And if you have a disorder where your tight junctions are not holding properly, that's bad. Things can get in that shouldn't and get out that shouldn't. So desmosomes are short proteins in the plasma membrane. We call them cadherins that act as spot welding. Okay, so these desmosomes, they kind of function like plasmodesmata, but they don't have a pass through. They're just welding the actual plasma membrane together. And so they're going to join adjacent cells in tissues that need to be able to stretch, because you can get basically you can get movement between these, and it allows for it to stretch outwards if necessary. And so some tissues need to be able to stretch, and like the heart and lungs and muscle. So desmosomes are necessary to weld these tissue cell membranes together, but allow for stretching at the same time. These are only present in animal cells. Okay, so gap junctions. Now, gap junctions are similar to the plasmodesmata in that they don't really hold the cells together, but they do allow for information to pass through them. So gap junction is a protein line pore that allows water and small molecules to pass between adjacent animal cells. So they're going to resemble the plasmodesmata, but they form channels that allow ions, nutrients, and other materials to move between them. Gap junctions are going to develop when 6 proteins, we call them connexins, form an elongated donut-like structure, a connexin, in the plasma membrane. And then when connexins of adjacent cells are aligned, that completes the channel. So one forms on the top, one forms on the other cell, and when they line up, it allows for movement to pass through of ions and different nutrients and that kind of thing through the animal cells. All right, so here's just everything all in one picture. So you can see the tight junctions are going to prevent fluid from moving across the different layers of cells. And then you have the desmosomes here that are going to allow for expansion in tissues that need it, like heart and muscle and lung tissues. You've got intermediate filaments shown here. Those intermediate filaments are helping give structure and hold organelles in place. You got the gap junctions down here that allow for information to pass through from cell to cell. And then you've got some ion channels that are specific to specific ions, like aquaporins only allow water, that kind of thing. So there are sodium sodium channels that only allow sodium. There's and potassium, that kind of thing. So there are ion or small molecules, and those can be considered just gap junctions as well. So yep. And now this is the study guide for chapter 4. So you need to be able to go through the study guide and answer these questions and be very familiar with all of these questions. If you have questions of your own that you don't really understand how the answer is applied to these, please reach out to me by email and we can get on a Zoom and go over it. I had one student do that with me for the past test, go through their study guides, and they did very well on the test. So if you want extra help, I'm here for you. All right, so now we're going to just jump right in to chapter 5, and we're going to start with membranes and transport. So plasma membrane functions. They're going to define the outer border of the cell and organisms. That's what a plasma membrane does. It defines the outer border of all cells and organelles. So it manages what enters and exits the cell. It's going to receive external signals that initiate interior cellular responses. It's going to also adhere to neighboring cells. So that's what the plasma membrane does. So we call this plasma membrane a fluid mosaic model. Okay. The reason we do call it that is because this membrane, this phospholipid bilayer, is able to be very fluid. Okay. It can move, and we need that to happen so that your cells can move through different areas of the body. So it's very important for movement. So we're going to break down this mosaic component of phospholipids, cholesterol, proteins, and carbohydrates that are able to give this membrane its fluid characteristic. So let's talk about the phospholipids in a little greater detail. We've already been over phospholipids, but it's important to remember that phospholipid is a type of fat. You've got these fatty acid tails down here, and these fatty acid tails are hydrophobic. They do not like water. They do not like it. Okay, so hydrophobic, do not like. And then you've got this head group up here that we call a hydrophilic head group. It loves water. It loves it. Okay, so that's supposed to be a heart. That was a horrible heart. We're just gonna let's see if I can do this better. I can do it better. Whoop. Yes, loves water. All right, so and it's comprised of this glycerol backbone here, and then it's gonna have a phosphate group attached. That's what gives it its hydrophilic nature, because you've got this big negative head portion up here, and then you've got a positive area down below, making this head group polar, and polar things love polar water. Okay, that's why it's an amphipathic, amphipathic, whatever you want to call it. Nature, it's what that means is it has both water loving and water hating. Okay, that's what, and I made that slide really congested. So basically all you got to remember is we love to touch water up here. We do not want to touch water down here. All right, so that's what gives it its amphiphilic or amphipathic nature. All right, so what exactly is a phospholipid bilayer? So it's when these phospholipids, singular phospholipids that look like this, arrange themselves in such a way that it creates two layers. So you've got a layer here and you've got a layer here, and the tails orient themselves in the middle because they don't want to touch water, because water's up here usually and water's down here usually. So it forms this layer, a bilayer, in which the head groups can touch the water, but the tails stay away from it. Okay, so they can do this, and it will form a sphere. And so if you imagine this as a cell and you were to blow this up, all the stuff inside the cell just lives inside here, right? And then outside the cell is everything else. So that's how cells are formed. They form this lipid bilayer that encases itself all the way, leaving a hollow inside where the cytoplasm is. And then you, of course, have your nucleus located here in the middle. But everything outside can be an aqueous environment as well. So this is that thing that we need to get straight in your head. Hydrophilic. The easiest way to think about this is thinking about water. So you've got an oxygen group up here connected to two hydrogens. We've talked about how the electrons stay around the oxygen way more often, okay? And when they do that, it gives a negative characteristic up top where, because the electrons are being drawn away from the hydrogens down here, it gives them a partial positive nature, meaning that you've got an overall negative up here and an overall positive down here. That's polar. That is being polar. When you've got a negative and a positive in the same molecule, that is polar. So like okay, stuff that is like each other likes to be around each other. Okay. So these tails in here, they are non-polar. They are not polar. So they don't like being around polar water. But the head groups are polar and they like being around polar water. So hydrophilic means that it loves water. That's polar. Water's polar. Therefore, hydrophilic heads are polar and they're going to face outwards to touch the water. Hydrophobic, you're fearful of water. You hate it. And that's non-polar. That's the tails. They face inward. They do not like touching water. Okay, so you got to get this concept straight. Hydrophilicity, hydrophobic, you need to understand what is polar, what is non-polar. Water, polar. And that kind of explains everything else. Okay. So membrane proteins, these can get pretty complex, but we're going to keep it simple. Proteins are going to be the second major component of this membrane. They're located all through it, on top of it, inside it, everything. They function as transporters, receptors, enzymes. Remember, enzymes help speed up reactions, that kind of thing, or a catalyst of some kind, or they can be used in binding and adhesion to other proteins, other molecules that they need to grab onto, that kind of thing. So let's talk about the different types. There's what we call an integral protein, and this is going to be integrated completely into the bilayer. Okay, so integral proteins integrated completely into the bilayer. This right here is an integral protein. This right here, because it's all the way through the bilayer. So if it's integral, it's all the way through. This one right here, integral protein right there. Where's another one? Oh, here's another one, integral protein. Okay, so, and then you can't really, these are kind of buried, but these are going all the way through back in there. Those are integral. Now you can see this one right here doesn't go all the way through, right? This one right here doesn't go all the way through. So there are proteins that are not going to be integral, and they're called peripheral. So peripheral proteins are only on the surfaces, either the interior or exterior surfaces of the cell is peripheral proteins. So integral all the way through it, peripheral on the surface. All right. So let's talk a little bit about integral membrane proteins. Integral membrane proteins have one or more regions that are hydrophobic, composed of hydrophobic amino acids, and others that are hydrophilic. So if I were to ask you a question, where are the hydrophobic amino acids of integral membrane proteins located? Well, that they can only be in one place. If this is the phospholipid bilayer right here on both sides with their polar head groups all the way down. I'm just going to draw this real quickly. And then they're polar, right? So and I said, where are the hydrophobic amino acids that make up the integral protein, where are they located? Well, they got to be near the hydrophobic tails, right? That's like stuff likes each other. When it's the same, it likes each other. So the stuff that's in the middle here, let me change colors to, we'll go blue. The stuff that's in the middle, all of that is comprised of amino acids that are hydrophobic. That's why they're inside. Now, these little whippy tail-like, let's do a different, we'll do green. These whippy tail-like structures that are out here, well, they're out in the aqueous environments because there is water and an aqueous environment inside and outside of most cells. So they have to be made of hydrophilic amino acids. Okay, so that they're on the outside or the inside. They're basically outside of the membrane. So this is a hugely important concept for you to get, is that proteins are made of amino acids. Some amino acids are hydrophobic. Some amino acids are hydrophilic. The ones that are hydrophobic will make up the part of the protein that lies inside the membrane, because that's where the hydrophobic tails are of the phospholipid bilayer. Okay, so all those that I just drew, those are all hydrophobic amino acids. All these beta pleated sheets that are in the middle right here, all of those are comprised of amino acids that are hydrophobic. Okay, all these little wispy ends that are out here on the outside, those are comprised of hydrophilic amino acids. Okay, so all that is hydrophilic. They love water, so they can be in contact with it. All right. So here are, and my thing messed up a little bit, but basically these are the non-polar amino acids, and down here you've got the polar ones. Okay, so you've got negatively charged groups and that kind of thing. And I had a slide previous that it doesn't cut it off like this, so go back to that slide, but to look at these. But there you got non-polar, and then you've got polar groups right here. So non-polar and polar, and then some that are negatively charged R groups allow for some polar type characteristics as well. So, all right, carbohydrates. So they're going to be the third major component, and these are oligosaccharide carbohydrates. They're located on the exterior surface of the plasma membrane. They're going to be bound to either proteins forming glycoproteins or to lipids forming glycolipids. But their function is in cell-to-cell recognition. And this is an example. This is like your blood type: A positive, B positive, AB negative, all that. So the oligoproteins, these right in here, these are glycolipids that are in yellow. They're going to be necessary for cell-to-cell recognition and attachment as well. So that's what oligoproteins do. Let's talk about membrane fluidity and how that actually occurs a little bit more. So the membrane has to be flexible, but it doesn't need to be so fluid that it can't maintain its structure when it needs to. So fluidity is going to be affected by the phospholipid type. So phospholipids with saturated fatty acids can pack together more closely, and we talked about that. Phospholipids that have saturated, their tails are straight. They have no kink. Unsaturated fatty acids are going to have a straight and usually one that's kinked. Okay. So that is an unsaturated because there's a double bond that causes the kink, at least one double bond. All right. So the ones that are saturated, they're going to pack more closely together than unsaturated fatty acids. Therefore, saturated fatty acids are going to be much more rigid. So temperature is going to play into this. Cold temperatures are going to compress molecules and make membranes more rigid. So cholesterol, though, is going to help with this. It's located within the fatty acid layer, and it's going to act as a fluidity buffer. So this link down here gives a great breakdown of that. I'd highly suggest going watching that YouTube video. But cholesterol acts as a buffer to keep the membranes fluid when it's cold and not getting too fluid when it's hot. So up here, cholesterol proteins are going to kind of look like this thing right through the middle right here. So but yeah, let's see if there's oh, here, no, sorry, these are the cholesterol. I'm sorry, I didn't mean to point that out wrong. These are the cholesterol right here. So you can see that they're embedded into the bilayer, and they actually allow for that buffering. All right, so this is there's going to be an asymmetry to the membrane. Plasma membranes are asymmetric. Their inner surface differs from their outer surface. So, for example, interior proteins anchor fibers of the cytoskeleton to the membrane. Exterior proteins are going to bind to the exterior extracellular matrix. And then glycoproteins bind to substances the cell needs to import. So these are those glycoproteins, and they're going to be reaching out like kind of like hands that'll grab onto things that the cell needs to bring inside it. But so you need to just kind of realize that there is a difference between the interior and exterior of the cell, and they have different functions. The interior is going to have different functions than the exterior of the cell. So there's going to be this selective permeability with the cell. The plasma membrane is selectively permeable, which means that it allows some things to pass through and not others. This feature is going to allow for the cytosol solution to differ from exterior fluids as well. So the interior fluid is different from the extracellular fluid. It's still mainly aqueous solutions inside and out, but they do differ. So example, all cells are going to maintain an imbalance of sodium and potassium ions between the interior and the exterior environments. So, and then you can transport across these membranes is going to be achieved by either a passive or an active transport. So passive, no energy is needed. Active requires energy to pass material across the membrane. All right, so now the simplest means of transport is diffusion. It's going to be a passive transport, and it occurs when substances from an area of high concentration move down its concentration gradient. That means it goes from an area of high concentration to less concentration. It wants to kind of distribute evenly. So this is going to occur through the lipid bilayer. Net movement ceases once equilibrium is achieved. So once you get to equilibrium, it's done. So think of it like this. You've got this bilayer right here. This is the bilayer. This is, we'll call this the outside of the cell. We'll call this the inside of the cell. And we've got all these molecules up here outside the cell, and they're going to start to diffuse through into the cytoplasm, and over time you get an equal distribution, and that's where diffusion will stop. So only small nonpolar molecules like O2, CO2, lipid hormones can diffuse through the biological phospholipid bilayer. All right. What are some factors that affect diffusion then? So these are going to be concentration gradients. Actually, you know what? We're going to go ahead and stop here and pick this back up for Friday's lecture. But if you have questions on any of this, you remember to get credit for today's lecture. You need to have attempted chapter 4 if you didn't already get 100 on it. And you need to go ahead and attempt chapter 5. I know we're only not quite halfway through with chapter 5, but go ahead and get on it anyway, so that you start getting some practice with it. But to get credit for attendance, you must attempt chapter 4 if you didn't get 100 last on Monday, and you need to go ahead and attempt chapter 5 as well, since we're already starting chapter 5. All right, have a great day.